So today we are talking about polar and non-polar molecules. So this is in the context of AS biology. Please don't take this lesson into your chemistry class and quote it as verbatim. This is the understanding of polar and non-polar molecules. It is the same basic concepts, but we're simplifying them and understanding them in relation to biology rather than chemistry. So when we talk about polar and non-polar, we're talking about charges. Okay, so we've got two options. We have ions and we have dipoles. So ions are our molecules that have full charges. So we could have a plus, or we could have a minus, or we could have a 2 plus, or a 2 minus, or we could even have a 3 plus, or a 3 minus, and so it goes on. So these guys have a lot of charge. So a full plus will stick to a full minus, or a full 2 plus could stick to 2 full minuses. And so we can start getting interactions between ions. We've got a lot of charge going on, so we have a relatively strong bond. When we're talking about dipoles, this is where we're talking about delta plus and delta minus. Okay, so the delta stands in for just a little bit. So these ones have a little bit of charge. So if you think back again to your ions versus your dipoles, when we're talking about ions, we're talking about that whole thing between the nucleus of the atom and the electrons. So here, sorry, I'm in the wrong place. Here, we're talking about gaining or losing electrons. So a full charge. Here, we're just talking about not sharing electrons fairly. So it's a lot less of a charge difference when we're talking about a dipole as opposed to an ion. So ions come up in AS biology, but dipoles come up quite a lot more. So what we're going to do is we're going to digress a little bit now and talk a lot more about dipoles and how they affect the things that you're going to be studying in AS biology. How do they affect in water, in biological molecules when we're building this whole story of hydrogen bonds. So ions and dipoles. Let's talk about how we get one of these dipoles. Okay, so we know, I hope, that when atoms are bonded in a molecule, they share electrons. Okay, so atoms in a molecule share electrons in what we call covalent bonds. Okay, so if we draw this really badly, in an understanding for chemistry, we have the nucleus of one atom, and we have the nucleus of another atom, and then we have their electron orbital shell thingy whatsits that you learn all about in detail in chemistry, right? Now, what happens in a covalent bond is the electrons go between both of them, so they share. Okay, so these electrons are flying around and they're being shared between the two atoms that are making up the molecule. So that sounds all very fair, all very balanced, we are sharing. But here, what happens is that not all atoms are created equal. We have bully atoms. Okay, and what that means in the context of charges, it means they have a bit of more of a pull on that electron. So rather than zipping around both of these evenly, it's like pulled more towards one side. Right, so coming back to this. When one atom has a stronger pull on the shared electron, the electron isn't balanced between the two. So we have a slight charge difference, our delta plus and delta minus. And that creates the dipole, which is our delta plus and delta minus. So one side will be delta plus and the other side will be delta minus. So we can sort of understand it in a sort of biological fashion, ladies and gentlemen, not a chemistry fashion. Instead of being even, we now have our electron, being very uneven. So here we would have delta minus and here we would have delta plus. So each time you see one of these, then we're getting a charge difference. We're getting polarity. So what we call here, this is a weakly polar molecule. So your ions are your polar molecules. Your dipoles are your weakly polar molecules. Right, so how does this then come through into biology? If we then take it to talk about dipoles in biology. Okay, so there are three big ones to be aware of. You've got your OH, your C double bond O, and your NH. Okay, so each of these is a covalent bond. So little note, solid line indicates a covalent bond. So we're sharing electrons. Okay, if we have two double lines, it's a double bond. If it's one line, it's a single bond. So we've got bonds going on here. Our C would actually have two other bonds because C has to have 4 bonds. O2, H1, N3. Okay, right. Anyway, we're getting very chemical there. In these dipoles, who is who? Well, it's nice and easy to remember. Your O's and your N's, oh no, are delta minus. So that makes the other pieces delta plus. So this will be delta plus, this will be delta plus, and this will be delta plus. Now these are pairings that we come across a lot in AS biology. And what happens is that you get a hydrogen bond forming between a delta plus and a delta minus. So we're going to use water as an example because he's nice and easy to draw. But the same thing works wherever you've got any of these dipoles. So water is H2O, so 2 little H's and a big fat O. And remember, now we've got dipoles because we've got an O bonded to an H. So the O's are going to be delta minus. They're the bullies, they're stealing the electrons. And the H's are going to be delta plus. So same like in magnets, same like in ions, opposites attract. So O to H. But see here, we've used a dotted line. And that dotted line is indicating a hydrogen bond. So opposites attract. Dotted line indicates a hydrogen bond. So one little hydrogen bond, one delta plus to one delta minus is really not that much. But when you get lots of them, it's rather like Velcro. One little hook and one little loop is not strong. But when you get a whole lot of hooks and a whole lot of loops, then it's really strong. So hydrogen bonds affect all sorts of things with the features of water, which you study. But if you think about where these all appear, there's a whole lot more. Okay, so your OH is found in water. It's also found in glucose and many other carbohydrates. Okay, all three of these are found in proteins. So now you're getting to maybe understand why hydrogen bonding forms such an important part in protein structure. We've got lots of dipoles going on in our amino acids. And when you have lots of dipoles going on, you have lots of opportunity for hydrogen bonding. And when you have lots of hydrogen bonding, you increase stability of the bond. Okay, the other place you get these ones in, all of them, is your nitrogenous bases. Okay, so in your nitrogenous bases, remember this is structure of DNA, you've got hydrogen bonding. So we've got water, we've got glucose, we've got proteins, we've got nitrogenous bases. The fun and games comes in when we get to lipids. So we're going to talk more about those. So we're now happy with everything where it is. Okay, so we're going to talk about lipids. We're just going to talk a little bit more about water first. Okay, so upping the game with regards to water. So we have said that water has a dipole. Because of delta plus, delta minus from the OH bond. So what this means is that we have a weakly polar molecule. We have, therefore, hydrogen bonds. But also remember that we've said that opposites attract. Okay, so plus to minus when you have polar. When you have polar versus non-polar, we have avoidance. So water is polar. It can associate with other polar molecules. And this is where it ties into that whole sort of dissolving and being in solution and acids going to their pluses and their minuses and hydrogen ions and all of that nonsense that you cover in chemistry. Not nonsense, but you know what I mean. So you can dissolve something because you've got this mixability. Okay, so everybody joins in the game with this polar molecule story. This is where we get the idea of hydrophilic from. Hydrophilic, liking water, okay? So it means that those are also polar molecules. They can be dipoles or they can be full on, like an ion with a full charge difference, okay? But that would make them hydrophilic because they're happy to associate with that dipole in the water. What's the opposite? The opposite is a non-polar molecule. Here there is no charge difference. Is hydrophobic, water hating. So if you don't have a charge, then you don't want to have anything to do with even a weak charge. You just want to be completely separated from that. So that's where we talk about oil and water don't mix, okay? Oil is hydrophobic. It doesn't have any charge. And that leads us beautifully into touching a bit now on how this whole story deals with lipids and their special situation. So, lipids in general are nonpolar. Phospholipids are special. So we're going to break lipids down into fatty acids and phospholipids and the phosphate head. So we have fatty acids. Think of that fatty acid tail, right? We do it with a zigzag and it's full of only carbon-hydrogen or carbon-carbon double bonds. This is no dipoles, which makes them nonpolar, which makes them, therefore, hydrophobic. Okay, so you start from the bonds, then you think about polars or ions, then have we got, dipoles or ions, then are we polar or nonpolar, then are we hydro whatever. If we think about the phosphate head, so really this is actually phospholipids, because they're the interesting ones. Does help if you spell it correctly though, doesn't it? Phospholipids, fatty acid tails, and a phosphate group. When we're talking about triglycerides, we're stopping at this nonpolar. Okay, when we come into this phosphate group, this one is an ion. This has a full ion charge. So we're just going to draw it a little bit. Remember, we have our glycerol, we have our ester bond. That should ring a bell, C double bond O. Then we have our tails, and then we've got CHs. And here we have an O, and then we have a P, and then we have a whole lot more going on. So up here we have phosphate ion and more. Because I am not drawing all those details and it's not something you need to know. Okay, the important thing is that you have a phosphate ion, but you also have these C double bond O's here. So all joined together, our hydrophilic head, we take to here. So this is our polar hydrophilic head. And these are our nonpolar hydrophobic tails. So this is where we get that difference in charge, and that is how we get the structure of our cell membrane and why we can have hydrophilic and hydrophobic within one molecule, because it's regions. Remember that these are very long. So it's not just a case of a couple of carbons and a couple of hydrogens. Okay, those fatty acid tails are really long. So you already have a proper piece of the molecule, which is nonpolar and hydrophobic. So your cell membrane structure is all about like with like. So those hydrophobic tails want to be together. And your hydrophilic, that's an O, there we go. So you got a hydrophilic core of the tails, you got a back to front. You got a hydrophobic core of tails, you got a hydrophobic outside and inside surface of your membrane. Okay, and that is then sort of tying everything together. So if we just recap a bit, we've got from the very beginning, we've got charges. Okay, we can have full-on charges, as in our ions, where we have pluses and minuses, or multiples, pluses and minuses. And we have weak charges, which is our dipoles, our delta plus and our delta minus. So when it comes to the ions, it's big, it's loud, it's in your face, it's obvious what the charge is. When it comes to dipoles, it's less obvious. When it comes to ions, we've actually lost a whole electron or gained a whole electron to do that charge. When it comes to dipoles, it's about not sharing quite evenly. So one atom is pulling that electron more towards it. So it's getting more negative charge, meaning the other one's getting less negative charge. So it's slightly positive. That's why we use the delta plus and the delta minus, because we're not adding or gaining a full electron, we're just not sharing properly, so we only have a little bit of charge difference. When we get into AS Bio, that little bit of charge difference is really important in quite a lot of things. So there's your 3 bonds that you've got to be aware of, your NH, your C double bond O, and your OH. Each of those has a dipole, and when you have a dipole, you have that ability to make a hydrogen bond. And hydrogen bonding is critical for a lot of things. So think, for example, if you look at water, your features and properties of water are dependent on your hydrogen bond. If you look at carbohydrates, then cellulose, the whole strength of cellulose depends on hydrogen bonds. If you look at proteins, then you've got hydrogen bonding again. Actually, I should be writing this down, shouldn't I? Let's just make there. Okay, so hydrogen bonding. Places to note. So hydrogen bonding, we talked first about water, properties of water. So this is hydrogen bonding highlights. Where we talk about carbohydrates, we really want to focus on cellulose. When we talk about proteins, now we're talking about levels of protein structure. We said there's lots of these dipoles in proteins, so therefore we have lots of opportunity for hydrogen bonds. So here we are talking about secondary, we're talking about tertiary, and we're talking about quaternary structure. Okay, then there's the side note of enzymes when you're talking about proteins and hydrogen bonding, because of course our enzyme substrate complex is held together by hydrogen bonds. So let's put that one in as well as a side note. So enzymes, your ES complex. When it comes to lipids, a little bit different here because it's not so much hydrogen bonding, it's actually polar versus non-polar. So we're talking hydrophilic versus hydrophobic. Then the other one for hydrogen bonding highlights has to be DNA. So base pairing. One is weak, many are strong. And that is so much of what happens with the structure and the protection of our DNA.